Recall that neurons get stimulated at their dendrites, and the stimulation at the dendrites causes what we call graded potentials. And these graded potentials can either eventually trigger an action potential or they can actually prevent an action potential from occurring. So, what we're showing here is a graph representing different graded potentials that might occur at the dendrites. Notice here we are at rest at negative 70. So during this time point, this neuron is not getting stimulated or inhibited by another neuron or another stimulus, such as a mechanical stimulus. Here we see there is a small depolarization that occurred. A small stimulus causes a small depolarization of the cell membrane. Recall that at depolarization, the membrane becomes less negative. But notice that it's not enough to reach threshold. On this graph, they are representing this dashed line as the threshold, negative 55. Another stimulus occurs, slightly larger, but still not enough to reach threshold. Here's a longer stimulus, but it also does not reach threshold. It isn't until this one here we see that the depolarization has reached threshold, and therefore that would be enough to trigger the action potential. These are all depolarizing graded potentials. Here we see what might occur if there is something inhibiting the neuron, such as an inhibitory neurotransmitter. So these are all examples of graded potentials, yet they are hyperpolarizing the membrane. To hyperpolarize, the membrane potential goes below negative 70 or more negative. So here's a couple hyperpolarizing graded potential. Here's a very dramatic hyperpolarization. All of these make that membrane farther from threshold, therefore it would make that neuron harder to get to threshold and cause an action potential. On this graph, we see the actual measurement of an action potential. So notice here we have a neuron, yellow representing its cell body and dendrites, green representing the axon. These are the little end bulbs. Let's imagine that right about here at the trigger zone we have an electrode, and that electrode is monitoring the membrane potential. During this period, in purple, the neuron is at rest. It's at negative 70. So if we refer to the key, that phase is the resting membrane potential. Voltage-gated sodium channels are in their resting state, and voltage-gated potassium channels are closed. Basically, these channels are all closed at this point. At the arrow, we see there was a stimulus at the dendrite. Could be mechanical, could be chemical. But whatever that stimulus is, notice it caused a depolarization. So the membrane is going from negative 70, less negative, towards negative 55. If we refer to the key, during that phase, there's a stimulus that causes depolarization, and it reaches threshold. If threshold is reached, notice what occurs. You have a rapid depolarization phase, shown in green. If we refer to the key, voltage-gated sodium channels open. That's what causes the depolarization phase of the action potential. Because when those voltage-gated sodium channels are allowed to open, you get a massive influx of sodium. Since sodium is a positively charged ion, you move towards positive charge. At positive 30, that's another key number to remember. At that number, we have closing of the voltage-gated sodium channels and opening of the voltage-gated potassium channels. This is going to cause the repolarization phase of the action potential. Notice in the key, voltage-gated potassium channels are now open. The voltage-gated sodium channels are inactivating, or said another way, they're closed. So now potassium, since it is at a high concentration on the inside, it's going to want to move out of the cell. So if you move a positive ion out of a cell, that cell is going to go back towards negative. So it goes back to resting membrane potential. Sometimes there's this after hyperpolarizing phase, but eventually we normalize back to that negative 70. So these are the key events to the action potential. I suggest you watching the animations that can be found on Canvas that will go over many of the important aspects of this process. Here's just a summary. This is a really good summary of the different structures of a neuron, as well as the function of each of those structures. So in review, dendrites receive the stimuli. Those stimuli are going to produce graded potentials, which are either going to be depolarizing graded potentials or hyperpolarizing graded potentials. The cell body basically is the area similar to dendrites. It can receive stimuli, but it's going to contain the nucleus. So this neuron, in fact, can get stimulated anywhere where there's yellow, the dendrites as well as the cell body. The junction of the axon hillock and initial segment is the location where the action potential will arise. And then the axon is going to propagate the wave of depolarization followed by repolarization until it reaches the end bulbs, where we have an inflow of calcium ions due to the voltage-gated calcium channels located in those end bulbs. And when that calcium influx occurs, we get exocytosis of the neurotransmitter from synaptic vesicles. So this is a really good summary of a lot of the important events that we've discussed. This is also a good comparison table, looking at graded potentials versus action potentials. I'm going to let you read through this table. Again, I would focus on where do they occur, what kind of channels are involved, do they propagate or not, what is the size? Graded potentials will vary in size, whereas an action potential is what we call all or none. So you will either get action potentials or you will not, but they're all going to be similar in size or amplitude. Duration, polarity that occurs, and then refractory period has to do with whether there is a method to prevent the depolarization from occurring immediately again. So it's present in action potentials. What will happen is the refractory period will prevent the action potential from going in the opposite direction. Not as important to us as some of these earlier characteristics.